So this is a way of considering basically biology, but from very much a genetics perspective. So this is actually something that we went through in the first chapter. This idea that you have genotype. Genotype gives rise to phenotype. Phenotypes interact with environments. In going from genotype to phenotype, you have something known as gene expression. And gene expression is definitely part of what molecular genetics is all about. Gene expression requires something known as transcription and translation, which we'll be talking about. The interface between phenotype and environment is ecology. This is something that we'll be getting to as well in a couple of chapters. And then you have feedback coming from the environment onto phenotype and then back to genotype. That's evolution, particularly natural selection. We'll be getting to that as well in a later chapter. But these same ideas, we can talk about them in slightly different ways. So when you're talking about genotype, you're more or less talking about genes. They're not 100% identical concepts, but you're more or less talking about genes. The genes are encoded in DNA, and certain viruses, they're encoded by RNA. The genes are then read. In the course of gene expression, you have a reading of the genes. And this reading involves transcription and then ultimately translation, processes we'll get to in a second. And this gene expression of these genes gives rise to the cells, gives rise to the bodies of the organisms, gives rise to the equivalent of phenotype. And then you have various kinds, in fact, you also gives rise to these various chemicals that we talked about earlier on. And then you have various kinds of interactions that go on between cells and bodies, between those organisms and ecosystems, and, or I should say abiotic components of environments to give rise to ecosystems, et cetera. So you've got various ways that you can consider these things. This is something we'll get to when we get to ecology. Cells and bodies, we've already talked about how cells work and the underlying biochemistry, but we'll be getting back to talking about organisms themselves when we start considering things like plants and fungi and animals. For today, what we're going to be concentrating on, or this chapter, the first half of it, is the genes and how you go from here to here in a genetic sense. So this is another way of representing the same ideas. In molecular genetics, there's something known as the central dogma. And the central dogma is a description of the flow of information as it occurs within cells. And this flow of information is shown right here. So basically, you have DNA, although again, in certain viruses, it's instead RNA. DNA gives rise to RNA. That's a process known as transcription. And RNA gives rise to proteins, which is a process known as translation. So you have essentially the storage molecule for information being transcribed into a more temporary storage molecule of information, the RNA, which is then read such that specific proteins are produced by cells. Now, these proteins, as well as these RNAs, together give rise to the development of organisms and give rise to their anatomy, their physiology, their behavior. All of this stuff is phenotype. The molecular genetics is interested or is concerned with mostly the part that's just in this line here. So we're going to talk about replication, which is the duplication of DNA. We're going to talk about transcription, which is the making of RNA. And we're going to talk about translation, which is the making of polypeptides. And then ultimately, the polypeptides get together to form proteins.